white shiny gives us WWSS. Violet dull gives us WW big S, big S. These are our parental ones that I have over here. You cross these, right? You cross these inbred or true breeding lineages, and this is your F1. So then we write out, we'll just ignore what we did. So we cross these two, then we say, okay, here's our F1. Our F1 is big W, big S, little s, right? And now we're ready to move on. They were then back crossed to the strain of white flower, shiny leaves. This one, right, we all of a sudden notice, okay, this is an F1, heterozygous. This is just like what's happening with the test cross. We're not actually doing a test cross, right? Why? Because we already know that this individual is heterozygous because we knew the parental lineages. But if we didn't, then this would be a classic test cross to see if this individual is heterozygous there. We also now know that test crosses can be used not just to test the heterozygousness, right, of that F1, but we also use it to test for crossing over occurring between genes that are on the same chromosome. And so here we have this test cross going on. Here it is, and we then get the following results, okay? In the F2s, we get how many violet dull, so big W, whatever, S, whatever, and how many do we get?
50.
50 of these. Big W, and now we see, okay, here's a recombinant genotype, right? Little w, S, whatever, little w, S, S. How many white shiny? So let's just do the parental ones first. 50 of the dominant. How many white shiny?
46.
46. We say, oh, look at that. We notice there's an overrepresentation. Well, let's look at the recombinant phenotypes first. The recombinant phenotype of shiny violet, how many?
12.
12, and of white dull, how many? 10? All right, so here we go. We would expect in a test cross for it to be roughly equal. We could even go back and do a chi-square test here to test if it is significantly deviating, just because we've done enough chi-square tests up until now, we can say, okay, yeah, we know that's going to be a pretty significant number, right? So we can just, pass over that. Okay, so now what do we do from this one? Now what's the next step that we do? We now have our observed types, right? If we're doing a chi-square test, we'd have that. We could then generate our expected types. But in this one, we don't even need to do that, right? But we do need to do what?
Divide the.
Yeah, we have to divide them up between parental. We can even in the beginning say, okay, we know this one's parental. We know this one's parental. We know this one is recombinant. We know this one is recombinant. So how many total individuals are there? We add that up. It's 8, 9, 11, 18. Is that what it is? 11, 18, is that what you got, Si?
118.
Yeah, that's what I meant, 118, sorry. 118. So we say n equals 118, and we want to see what percentage are recombinant. So we go over here, we add 12 plus 10, we get 22 divided by 118. This gives us 20%, 19%?
18.6.
18.6%. That is then telling us what? Again, this tells us a whole bunch of things based on what we've done. What is it telling us? Does it tell us a probability? What's that probability? If we're looking at this in terms of probability, what?
Dream 2 combination is okay.
Yeah, so the probability that in the generation of a single gamete, there's an 18% chance, right? 18.6% chance that recombination is going to occur. It also tells us that in the generation of 100 gametes, what percentage of them do we expect to be recombinant? 18.6. So that's kind of the same flip side of the same coin right there, but think about it in both of those terms. What other thing does it tell us? This extrapolation that Alfred made, what? Yeah, so then it's telling us the distance between those two genes, the actual physical geographical distance between those two genes, okay? And so, as you can see, so we kind of did all, we're not going to go through and answer all of those, but that's how you do those problems. Okay. So now, they